So I decided to break this down a little bit and give you a little bit of a break because that DNA section ended up being a little bit long. So this next section is going to talk about protein production and transport. So far we've talked about the nucleus, and we said that DNA is found in the nucleus. Now if we look at this DNA, we said that it codes for proteins. So there's different regions on the DNA that consist of bases. So we can put A, T, C, G, blah, 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 or G, A, C, T, blah, 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 blah. And those regions are called genes. So genes are going to be a region of bases on DNA that code for a protein. So if we look back at this picture of what DNA really looks like, it's pretty far back, you can see it's just a bunch of strands. So if we take one of these strands and we examine what is in that strand, it's really just a series of bases. And each particular series of bases, if we want to make a protein from that region, that's called a gene. So a gene will code for a protein. It's a region of bases on DNA that code for a protein. So how does our body know where all of these genes are located in that mess of DNA? Well, each gene has a start and a stop, a promoter region that happens before the gene, and a termination region that happens after the gene. So we have a start and a stop to each gene. So let's say we have our strand of DNA and we have our gene that's located here. How do we turn that gene into a protein? Now remember, this is all inside of the nucleus. We can't just take this and move it out of the nucleus in order to make a copy of this gene and in order to turn it into a protein. If you recall, our ribosomes are outside of the nucleus, and ribosomes are where we have to get to in order to make the copy and make the protein from our genes. So we have to think about, how do we get the information from the DNA in our nucleus over to a ribosome to actually do something with it? And I use the analogy of baking a cake for your aunt in Arizona. So here's our analogy. And here's what's actually happening in the cell. So you want to bake a cake, a protein. And the recipe, or the gene, is located in the cookbook in your DNA, which is located in your kitchen cabinet. Or your nucleus. Now you want to get that recipe to the counter so that you can actually read it and make your cake using the recipe. You want to get that gene sequence to a ribosome so that you can actually make your protein. So how do we do that? We have our nucleus and we have this gene that's somewhere inside of the nucleus. Well, let's just make a copy. If we copy the gene, we can produce a strand of genetic material known as messenger RNA. Imagine that this cookbook weighs 2,000 pounds. Are you really going to lift the 2,000-pound cookbook out of your kitchen cabinet to set it up on the counter? No. You're going to squat down and you're going to flip to the page of the book that actually contains the recipe that you want to make, and you're going to copy it down. And then you're going to carry that copy up to the counter so that you can start reading it to make your cake. That's what we do inside of the cell. Instead of taking all of the DNA out of the nucleus, instead we just make a small copy of the gene. And that copy that we make is not DNA, it's actually RNA known as messenger RNA. That now is the recipe that we can carry to a ribosome in order to produce our protein. So here I've drawn our messenger RNA, and we're going to take that messenger RNA to the ribosome. In order to produce a protein. So that messenger RNA is going to have to leave the nucleus through a nuclear pore and eventually get its way into the ribosome. The process of making this copy is known as transcription. So transcription is the process of making an mRNA copy of a gene. So all you've done is jotted down the recipe right now. You have a physical copy of the recipe, you're carrying it over to the kitchen counter so you can actually read it to produce your cake or to produce your protein. So now you or a ribosome read the recipe. So the ribosome is going to read the messenger RNA. And as you read the recipe, you add ingredients to make the cake. As your ribosome reads the messenger RNA, it's going to use tRNA, transfer RNAs, to add amino acids to the protein. This is what's known as translation. So when the ribosome reads the recipe or the messenger RNA and incorporates a sequence of amino acids to make a protein. So our messenger RNA was produced, it went to a ribosome, our ribosome read the messenger RNA and produced this protein strand. Now we have to get this protein that the ribosome has produced to its final destination, to where it needs to be in the cell. So looking at this picture, we have smooth endoplasmic reticulum and rough endoplasmic reticulum. Now, if the protein was destined for the inside of the cell, it would have been made by a free-floating ribosome, and it would just hang out inside of the cell after production. But we're actually going to talk about what happens when a protein is destined for the outside of the cell. Smooth endoplasmic reticulum is responsible for hormone production, lipid metabolism, and detoxification of the cell. The reason why we call it smooth is because it does not contain ribosomes. Anything that's produced in the smooth endoplasmic reticulum will eventually make its way into a vesicle, to the Golgi apparatus, and to the cell surface. But we're going to focus on what happens in the rough endoplasmic reticulum because that's the next step along our pathway of making our cake for our aunt in Arizona, or producing a protein to ship to the outside of the cell. So rough endoplasmic reticulum does contain ribosomes. And it's responsible for producing proteins to be shipped out of the cell. So proteins for secretion, or proteins that are going to be incorporated into the membrane. So we made our copy, our messenger RNA copy in the nucleus. That messenger RNA copy leaves the nucleus and it enters right into the rough endoplasmic reticulum. We have ribosomes here that are waiting to read that message and produce amino acids from it. Now, as soon as the protein strand is made in the endoplasmic reticulum, we have to have a way to transport it. The way that we transport substances from the endoplasmic reticulum to the next step along the way, the post office or the Golgi apparatus, we have this vesicle. So a vesicle is just a transport vehicle, and it transports substances around in the cell. This either means that it's going to transport them to the Golgi apparatus for processing, or it's just going to hold them inside of the cell. But a vesicle is a membrane enclosed structure that transports substances around inside the cell. So going back to our scenario here, we've made our cake in the rough, let me erase that. In the kitchen, which is the rough ER. Now we have to get in our car, or a vesicle, to drive it to the post office, or to take it to the Golgi apparatus for processing. The Golgi apparatus is responsible for processing, modifying, and activating the substances that were produced in the endoplasmic reticulum. So it packages and modifies substances to be shipped outside of the cell, kind of like a post office. So at the post office, you put an address label on a box, you wrap it up, and you get it ready for shipment so that it can make the trip all the way to Arizona. So the Golgi apparatus plays a major role in shipping substances out of the cell. Now from that Golgi apparatus, we have to get back into let's say a mail truck, or back into a vesicle to be shipped to our aunt in Arizona. To be shipped to the cell surface. Having foundational knowledge of where proteins are coming from and how they're shipped out of a cell is really important. This is how neurotransmitters are produced and released in order to stimulate your nerve impulse. Hormones are produced from the smooth endoplasmic reticulum, and they're stored in vesicles until they're signaled to be released. So this is all very important.